Hello everyone, welcome back to Biophilia, your all-time study mate. Today we are going to complete this chapter about cell, the unit of life. If you like this video and get some help, then don't forget to like, share and subscribe to our amazing channel Biophilia. So as we have completed to understand about what is cell, now let's get deep into to understand what does the cell have. So cell जो होता है वो एक component है जिसमें और बहुत सारे छोटी छोटी चीजें होती है. There are subcellular structures and these subcellular structures which have many more specific jobs to perform inside the cell. जैसे एक organ होता है पूरे body में, वैसे each and every cell have few components that are going to function their specific jobs. So those are nothing but called as organelles. So we are going to discuss in detail the different types of organelles. जैसे पूरे body को एक अगर structure में divide किया जाए, so you can compare one cell which is equivalent to the entire body and you can compare the organelles which are going to function similar to the organs of the body. So in that way, there are different types of organelles which come as the plasma membrane, cell wall, nucleus, endoplasmic reticulum, which is also called as ER, then Golgi apparatus, lysosomes, vacuoles, mitochondria, plastids, ribosomes, cytoskeleton, then centrosomes, centrioles, cilia, and even flagella. So all these are the cell organelles that we have to study in detail about in this video. So first is the plasma membrane, which also has another name as cell membrane. So cell membrane, आपको ये पता होगा पहले से that cell membrane or plasma membrane is going to be composed of lipids. So lipids is a kind of fat. So this lipid is layered in bilayer, यानी उसके दो layers हैं, and these two layers are not arranged anyhow. These are arranged as per the hydrophobic and hydrophilic nature. So there are polar heads and there are non-polar tails. So polar heads जो होते हैं, these are hydrophilic. Hydrophilic, what do you mean by hydrophilic? Hydrophilic, hydro means word that is used for water and philic means love. So the polar head is going to attract to water, it is going to be okay with water so that it remains on the outer edges. जबकि non-polar tails जो होंगे, these are hydrophobic. Hydro is water, phobic is, you can say, fear. So the person or who here the non-polar tails that get feared of water, so these tend to stay on the inner surface. So because of this kind of water loving and water fearing nature of this plasma membrane component, there is going to be nature like you can see here in the diagram that the lipids are going to have the heads, they will have the tail. So the heads are going to be hydrophilic and the tails are going to be hydrophobic. So the tails will be on the inner side whereas the heads will be facing on the outer edges. So in the human being, the cells, that is majorly we speak about the erythrocytes, that is RBCs. So RBCs का जो भी plasma membrane है, it is going to be having 52% protein and 40% lipids. So this composition is going to be important with the respect of MCQs. So this composition needs to be remembered. So for ease of extraction, the membrane proteins are being classified into two ways. So the first one is going to be the peripheral proteins and the second ones being the integral proteins. So peripheral proteins जो है, these are peripheral, as the name suggests, it is on the periphery of the membrane, that is they lie on the surface of the membrane. So here you can see in the diagram as well what is the location of peripheral membrane protein. ये ज्यादातर बाहर होते हैं, you can see them from the outer surfaces, whereas integral protein, integral part of the team or integral part of something, that is the main important deep buried into it. So these are partially or they can be totally buried in the membrane, and this will run through the membrane. Those are called as tunnel proteins. यानी कुछ हो सकते हैं जो आर पार है, like these are half in and out, don't know where to go. Some are going to be totally inside and some are going to be like, these will be like tunnels. एक side से start होंगे, एक side पे जाके end होंगे. So those are tunnel proteins. So these being buried into the membrane, these are not easy to remove and it requires treatment of detergent to remove these kinds of proteins. जबकि peripheral proteins बाहर के बाहर है, these can be comparatively easily removed out of it. So tunnel proteins are also called as transmembrane proteins. What are they called as? These are also called as transmembrane proteins. So अगर आपको ये नाम दिखे या tunnel proteins दिखे, anyhow you have to remember these are the integral proteins that run through the lipid bilayer membrane. Next is the nature of the lipid membrane, or you can say the plasma membrane, that is fluid mosaic model. So fluid mosaic model was proposed by scientists, that was Singer and Nicholson, in 1972. And this membrane, why does it name of fluid mosaic model? Because fluid है, because this membrane is not static, it keeps on moving, and because of the movement of this particular membrane, it is termed as fluid. So ये membrane को जब सिंगर एंड निकोल्सन ने notify किया था, it was to compare with the protein icebergs floating in the sea of phospholipids. So if you might have seen the movie Titanic, then you must have seen that how the icebergs are seen in the ocean. So बहुत सारा पानी होगा और पानी के some glaciers होंगे that looks above floating, कुछ होंगे जो इनका बहुत छोटा सा पार्ट दिखेगा, and some might be completely submerged inside the sea that you cannot see from the top view, and then the ship might go and crash. So these kinds of proteins are present in the entire ocean or sea of phospholipids. So the icebergs are going to be the proteins. Like we studied, there are two types of proteins. So these proteins are icebergs, and the sea is compared to the phospholipids. Then is the quasi-fluid nature of lipids, that is these lipids are going to allow lateral movement. So these proteins are not static, वो एक ही जगह पे रुके हुए नहीं रहेंगे. Since it is sea, it is fluid, so these proteins can also move, and this ability to move within the protein is measured into fluidity. So the name, that is fluid mosaic model. So how is the plasma membrane was described by these two scientists. The next is the function of plasma membrane. So plasma membrane, as we saw, it is the fluid mosaic model. So the fluid nature of the plasma membrane is going to be very important. So जैसे cell growth हो गया, so cell growth हो रही है, there is growth, so the plasma membrane should also grow along with it. So because of the fluid nature, it can allow growth into it. The formation of intercellular junctions, so दो cells को join करके रखने के लिए, so there is no producing gap. जैसे ट्रेन के डिब्बों के पीछे there is big magnets set up that is going to hold the two trains. So same way, this intercellular junctions are also going to hold the two cells together. Then secretions, endocytosis, cell division, all this is going to be because of the fluid nature of the plasma membrane. So this plasma membrane is also going to allow some kind of transport to the molecules. अब ये जो transport हो रहा है, it is going to be of three types. So the first type being the passive transport. Passive word होता है, that is you are not getting engaged into it. It is just normally happening. So that is passive transport. So plasma membrane यहाँ पे actively कुछ भी involvement नहीं दिखा रहा है, but since it is getting passed through it, the plasma membrane transport is included over here. So passive transport में there is movement without the use of energy, so you don't have to spend any sort of ATPs along this transport. So this kind of is going to include diffusion, facilitate diffusion, and osmosis. So osmosis is generally the term that is used for fluids, or you can say it is used for water transport. So that is osmosis. Diffusion of gases, you know very well कि अगरबत्ती एक कोने में लगाई पूरे रूम में महक होती है. So that kind of thing is diffusion. And facilitate diffusion is like the glucose transport is facilitate diffusion, wherein there's something is going to help for the diffusion, that is facilitate diffusion. The next is going to be about active transport. So active transport यानी यहाँ पे कुछ actively function हो रहा है. So this is going to be a uphill movement, यानी ये against the concentration जाएगा. जबकि diffusion कैसा होता है? It is always to the concentration. That is, the movement will be how? It will be from high concentration to the low concentration. But in active transport, it can be possible that it moves from low concentration to the high concentration zones. So this active transport, to move against of something, you need to spend energy in the form of ATP. So for that reason, the Na and K pumps are going to be present, which are going to help in the active transport of things. Last is about the endocytosis and exocytosis. So endocytosis में दो types होते हैं. Endo is the word that refers to inside. Cyto is going to mean that is cell. So cell के अंदर लाने की जो process है, the cis word is used for some kind of process. So cell के अंदर लाने की जो process है, that is endocytosis, whereas exo means going outside again cell, and cis is the process. So this way you have to break the terms and understand the meaning out of it. So endocytosis में दो चीजें अंदर आ सकती है. As you put something in your mouth, that is, you eat food, that is solid things, and you drink water or fluids. So that thing is also done by the cell, that is pinocytosis. पानी पीते हैं. So पीते is related to pinocytosis, that is cell drinking process, and phagocytosis, that is cell eating process. So ये अंदर आने की चीजें है. The fluids and the solids are coming inside the vacuoles, that is endocytosis, and exocytosis is throwing out. So what do you throw out of your mouth? You vomit. So that is nothing but cell vomiting process. So these are the some functions about the plasma membrane and different types of transports that is being performed by the plasma membrane. The next we see is the cell wall. अब cell wall जो है, it is not living, that is, it is non-living. जैसे plasma membrane थी, it was a living entity, it had some function to perform. But here the cell wall is non-living, it is rigid, and this rigid structure is outside the plasma membrane if present. It is not compulsory to have a cell wall. For example, cell walls are present in the cells of bacteria, fungi, algae, and plants. But if you consider animal cells, do they have cell wall? So animal cells have no cell wall, right? So cell wall is going to be absent in the case of animal kingdom or animal cells. So if you consider for the cell wall of funguses, so that is fungal cell wall, it is going to be composed of a product that is chitin polymer. So chitin polymer is going to be made up of NAG units, that is N-acetyl glucosamine, that is going to be present in the fungal cell wall. जबकि algal cell wall की क्या खासियत है? It is made up of cellulose, galactans, mannans, and minerals like calcium carbonate, whereas plant cell wall is majorly composed of insoluble polysaccharides. Majorly it is cellulose, and along with cellulose, in some parts it can be pectin, it can be hemicellulose, and even some kind of proteins can be present. So cell wall is the rigid non-living which is going to be helpful for giving shape to the cells. Shape maintenance के लिए it is going to be helpful. So here you can see in light green color, the thing that you can see is nothing but going to be the cell wall. So the functions of cell wall is easy. to remember कि ये non-living है, यानी this is not going to help in any kind of active transport. ये dead चीज है, like it is not living. So this is going to help to maintain the shape of the cells. It is going to protect from any kind of mechanical injury because dead चीजें है, it is going to be very strong enough. Since it is dead, it is not going to allow direct entry, so pathogens attack is waved off. It is also going to allow direct entry, यानी ये selective membrane नहीं है. It is not like plasma membrane where there is a selective transport. यहाँ पे जो भी चीज high to low जा रही है, it is going to be allowed to go inside. Right, and then it also helps in cell to cell interactions and also provides barrier to undesirable molecules. So moving to understand about the layers of the cell wall. Cell wall एक layer की नहीं है. During the process of making of cell wall, there is going to be different layers that are going to be formed one over the other. So the first one that we speak about is the primary cell wall. So primary cell wall is going to be the middle layer that you need to remember. It is the middle layer kind of once you classify here. So this middle layer is made up of cellulose. It is going to show growth, but it will diminish as the cell is going to mature. Whereas if you speak with the secondary cell wall, it is going to be the innermost thing, and this innermost layer is going to be made up of lignin and pectin, whereas the last layer, that is going to be the middle lamella. As the name suggests, it is middle lamella, but actually it is not the middle portion of the cell wall, but it is going to be the outermost layer of the cell wall. So that is the major thing that you need to remember. This is present outside to the primary cell wall, that is, it is the outermost layer made up of calcium pectate and magnesium pectate. So these are salts of calcium and magnesium. Then it is acting as a cementing material. Cement क्या काम करता है? दो bricks को जोड़ता है. So दो bricks को जोड़ने के बीच का काम, the gluing work is made by the middle lamella. So middle lamella, since it is made up of calcium pectate and magnesium pectate, when the fruits are raw fruits कैसे होते हैं? Raw fruits are going to be hard. So raw fruits, that is hardness, but once it is going to be ripe fruits are going to be soft. So during that time, the middle lamella is going to disintegrate, and because of that, the middle lamella disintegration, the fruits become soft when they are ripe. So here also you can see in the diagram with the color coding, the pinkish red color is the primary cell wall. The inner three green layers are first, second and third secondary walls, and the last blue one is the middle lamella. So these are the layers of the plant cell wall. Now we speak about the third thing, that is the endoplasmic reticulum. The ultrastructure of endoplasmic reticulum is going to show three major structures of it. So this endoplasmic reticulum, endo means inside, plasmic that is inside the cell, the cytoplasm is present, wherein this kind of organelle will be present with three ultrastructures, that is the cisternae, the second is tubules, and the third is the vesicles. So the cisternae are sac-like structures, bag जैसे है, which are long, flattened and parallel, and these are interconnected. They have sites for ribosome binding, and also because of presence of ribosomes, this will be active in protein synthesis. And as you know that ribosomes are present onto the endoplasmic reticulum, that makes up the rough endoplasmic reticulum. So cisternae will be maximum in the case of rough endoplasmic reticulum. But once the ribosomes are out of, or they are disattached to the cisternae, तो वो क्या हो जाएगा, मेजर कंपोनेंट तो राइबोसोम्स है. एंड वंस द राइबोसोम्स आर लूज्ड, व्हाट आर दे गोइंग टू कन्वर्ट इनटू ट्यूब्यूल्स. So tubules जो है, these are tube-like structures. They might be branched or they might be unbranched. So as the ribosomes are lost, they form into tubules. So these tubules do not have ribosomes. These will be made up of lipids and sterol synthesis. Because ribosomes nahi hai, that means protein synthesis will not occur. And when protein synthesis not occurs, it is lipid and sterol that is produced by the smooth endoplasmic reticulum. So ज़्यादातर स्मूथ एंडोप्लाज्मिक रेटिकुलम में ट्यूब्यूल्स मिलेंगे. And this is the product of cistern passing over to the cytoplasmic theory tubules. The third structure is vesicles, which is oval spherical membrane bound vascular structure. It is going to be having a single membrane, and it also lacks ribosome, and it only endoplasmic reticulum which is present in the spermatocyte. So specifically in the spermatocyte endoplasmic reticulum, the vesicles are going to be present. जबकि सिस्टर्ने है rough endoplasmic reticulum that is found in all the cells, and tubules are found in smooth endoplasmic reticulum which is also seen in all cells. The next is the types of endoplasmic reticulum. As we said that it is smooth endoplasmic reticulum, that is SER, and the rough endoplasmic reticulum RER. So smooth hai, that means there is no ribosomes, so ribosomes are absent. And as we discussed, the ultrastructure has tubules more in quantity. Then lipids and sterols are synthesized because ribosomes are not present. And what is the function of SER? If they are not producing proteins, then these are going to be helpful in detoxification of drugs, calcium uptake, that is released into the muscle contraction, and the product of RER passes from the SER. यानी जो भी proteins बनेंगे, they are going to transport from the smooth endoplasmic reticulum. But processing कहाँ से होगी? It will be from the RER that have ribosomes. They are many cisternae present in them. Protein synthesis is going to occur, and it will, after the ribosomes disattach, they will have the formation of smooth endoplasmic reticulum. So that is the two types of reticulum that you can see in the diagram as well. So nucleus के पास में, since nucleus is going to send the mRNA. Now what is mRNA? You will shortly understand about. So mRNA जब जाएगा, so mRNA, or you can say once majorly the rRNA. So R, that is ribosomal RNA, once it is going out of the nucleus, then it is going to be having the function of protein formation. So protein formation कौन करेगा? Ribosomes करेंगे, which are onto the rough endoplasmic reticulum. So rough endoplasmic reticulum is in close proximity to the nucleus, whereas the proteins that are synthesized needs to be transported out from the smooth endoplasmic reticulum. So it will be comparatively off to the nucleus. So here you can see the red dots that are present. These red dots are nothing but going to be the ribosomes, which are going to be absent onto the smooth endoplasmic reticulum. The next organelle is the Golgi apparatus. The name Golgi apparatus is because of the scientist who found, that was Camillo Golgi. So it is based on his name. So Golgi apparatus is present in almost all eukaryotic cells. Now why the name almost? Because it is going to have some exception. So exceptions याद रखना ज़रूरी है. That is, it is not going to be present in the sieve tubes, the RBCs, sperm cells of bryophytes and pteridophytes. They do not have Golgi apparatus. So Golgi apparatus या Golgi complex को plants में अलग नाम दिया है, that is dictyosome. And since it is Golgi apparatus or Golgi complex, it is going to have some different structures in it as well. So these are four, that is cisternae, tubules, vesicles, and vacuoles. So what are these? Let us see them in detail. So the cisternae, as we saw in the endoplasmic reticulum as well, these are flattened sac-like structures which have two phases. The first one is a cis phase and second is a trans phase. Now this point is very important to remember because यहाँ पे MCQ के chances ज़्यादा होते हैं. So cis phase is going to be having the formation phase, यानी जो भी तैयार होने वाला है that is onto the cis phase, and जो भी mature होगा to come out of the Golgi apparatus, that is the trans phase. So vesicles are going to be coming nearer to the endoplasmic reticulum. They will enter into the Golgi complex from the cis phase and they will process, get packed with lipids and sterols, and they will come out of the trans phase. The next thing is about the tubules, which are small tubular structures that arise from the periphery of the cisternae. So cisternae के पास में से ये tubules बाहर निकलेंगे. Then the vesicles are large vacuolar structures which are present at the end of cisternae. So here you can see about the newly forming vesicle, or you can see the secretory vesicle. These are the vesicles. And vacuoles, you know vacuoles, that is the majority of the plant cell, that is large vacuolar structure arising from the maturing phase, is going to be called as a vacuole. The next functions of Golgi apparatus, that is, as we saw that यहाँ पे packaging होती है. So it is kind of a packaging station, यानी जो भी चीजें endoplasmic reticulum synthesize करेगी, proteins, so these are generally directly sent into the cytoplasm, but lipids and sterols need to be packed. So majorly the processing and packaging of it might be proteins, lipids or sterols that comes from the endoplasmic reticulum is completed at the Golgi apparatus. So it is very important to understand that the nucleus, the endoplasmic reticulum and the Golgi apparatus, these are very much closely related to each other and these are placed in close proximity because in सबका function कहीं ना कहीं एक दूसरे से जुड़ा हुआ है. So after the vesicle formation, vesicles के अंदर, if there is some kind of enzymes present in it, that becomes the lysosome. So lysosomes are also synthesized with the help of Golgi apparatus. Even the cell wall material synthesis is completed by the Golgi apparatus. Formation of plasma membrane, because here there is going to be lipids present. So lipids coming from here. The root cap is going to be very highly rich in Golgi bodies. Forms the acrosome of the sperm. So sperm structure, once you see, the upper head-like structure is going to be majorly the Golgi body entirely. Then formation of cell plate is also because of the Golgi apparatus secretion. The next is the vacuoles, which are membrane bound spaces. So this is very important, that is these are membrane bound, because one thing you will also see, there are some other structures that looks like vacuoles but these are not membrane bound. So vacuoles are the kind of storage unit, you can say जो भी सारा डब्बे जैसे डब्बे के अंदर you store anything. So these are kind of foldable dabbas. Whenever you need anything to store inside, that it might be the excretory waste or it might be some storage spaces, all that thing will be stored inside the vacuoles. So if you consider the case of plants, the 90% area of the cell is going to be covered by the plant vacuole. And inside the vacuoles, what do the plant cell keep? They keep the cell sap. So अगर ऐसे कभी हुआ है that you try to cut a part of the stem and some fluidy material that comes out of it, या आपने leaf को pluck किया है and जहाँ से pluck किया वहाँ से कुछ white fluid जैसा निकल रहा है, that is nothing but kind of the cell sap. So that is stored inside the plant vacuoles. Then it also plays an important role in endoosmosis and exoosmosis, because जो भी चीजें अंदर बाहर होने वाली है, so if it is coming inside, it is going to be collected inside a vacuole. If it is going out, then it is thrown out from the vacuoles. So in that case, these are important. Then the movement of cytoplasm that occurs around the vacuoles, जैसे for example एक सेल है, ये सेल के अंदर कहीं वैक्यूल है, तो वैक्यूल के अराउंड अगर साइटोप्लाज्म सर्कुलर मैनर में मूव कर रहा है, then that circular movement around the different vacuoles is going to be called as cyclosis. Now vacuoles being, having different kinds of roles in the cell, these are not a single type of vacuole. इनके भी कुछ types होते हैं. Like for example, contractile vacuole, which is going to help in excretion for amoeba. जैसे amoeba, it is a single cell animal, and because of that you can say excretion, जैसे पूरा excretory system होता है in the case of larger animals. अमीबा के पास सिर्फ एक कॉन्ट्रैक्टाइल वैक्यूल होता है, which is going to help in entire excretion process. Then gas vacuoles होते हैं, which have gas present in it, and because of the gas, they are going to provide buoyancy, that is floating movement provide करेगा. So for example, if you consider the lotus leaves, so the free floating plants, or say the lotus leaves, these are going to have a lot of gas vacuoles. Why? Because they have to provide buoyancy so that they can be free floating and they can be on the top surface without being attached downwards. Then food vacuoles, as the name suggests, food, that means food engulfing, that is in the case of protists, is going to be done by the vacuoles. Then is lysosomes. As I said about lysosomes, these are tiny spherical sac-like structures formed by the Golgi apparatus. But Golgi apparatus जब इन्हें बनाता है, these are kind of vesicles. But inside these, sac-like structures, there are going to be different hydrolytic enzymes. These hydrolytic enzymes make them special, called as lysosomes. These are again single membrane bound. Again, that means they have membranes. So now in future slides, you will understand that there is one organelle which is not membrane bound. So do remember that only that is one that you need to remember, that is, it is not having a membrane. There are some who have a single membrane. There are again few organelles that have two membranes. So these are the things that you need to remember. Being lysosomes, they have hydrolytic enzymes. So digestion के लिए जैसे पेट का shape change होता है, like sometimes when it is empty, it is smaller, and once it gets full, it is going to stretch out to occupy more space. So movements होते हैं in this shape, that is showing polymorphism. So there are hydrolytic enzymes. So these enzymes are going to get activated at the acidic pH. That is, you can say from 0 to 7 pH, you can say it is activated. So protease, protein related. So these are digesting proteins. Lipase for lipids, and nuclease for nucleic acids. So again, the types of lysosomes to be understood. The first one being primary lysosomes, which are called as suicidal bags. So these, when are formed from the Golgi body, they have inactive enzymes. But अब ये जब active हो जाते हैं in enzymes, they become secondary or heterophagosomes. So when the primary lysosome gets attached to a phagosome, it is called as a secondary lysosome. Third type is the residual bodies, यानी जब digestion complete हो जाता है with the help of the hydrolytic enzymes, the substance which was present in the secondary lysosome, now it has entirely digested, और जैसे undigested material उसके अंदर present है, which is moving out for excretion, that is with the help of exocytosis or enphagy, that is the residual bodies. Next is the mitochondria. It is also called as powerhouse of the cell. Why it is called as powerhouse of the cell? The reason is going to be the production of energy. So ये energy produce करता है in the form of, you can say, ATP, that is adenosine triphosphate. So it is a double membrane bound. So again, this is an important question. Double membrane hai. The previous ones that we studied, these were single membrane. It is a sausage shaped cell organelle which has outer membrane and inner membrane. So outer membrane जो है उसके पास 60% protein है, 40% lipids है, whereas for the inner membrane there is 80% protein and 20% lipids. So this composition needs to be remembered. किसके पास ज़्यादा protein है और किसके पास कम है. So the foldings are going to be present in the inner membrane. Outer membrane smooth है, but the inner membrane is going to be wavy or it is folded, and the folds inside the inner membrane are called as cristae. Now inside these foldings there is going to be electron chain running, and this electron chain is very important for the energy production. So ATP generation जो होगा, it is going to be because of the electron chain that runs inside the inner membrane. So this mitochondria, it has the double membrane, as well as very important to understand, it also has a DNA along with ribosomes present inside. So since it has a double circular DNA along with ribosomes, it looks like, जैसे आप कंसिडर करो एक पूरा का पूरा सेल है, वो सेल के पास न्यूक्लियस होता है, and in that nucleus you are going to have DNA, right? But here you can see that उधर एक sausage shaped का organelle है, and once you take this organelle out, you are going to see the sausage shaped having the wavy structures, the inward foldings, और उसके अंदर भी you can see a dsDNA. So cell के अंदर एक और cell जैसा structure है. So that is the reason it is called as the cell inside cell. So again, this term is important to remember. So how is a new cell formed? So it can be formed by fission. So similarly, mitochondria is also formed by the fission. So one mitochondria splits into forming two new mitochondria. So the next is the plastids. These plastids are going to be again somewhat similar to mitochondria. Why they are similar? Because again these are going to be double membrane. इनके पास भी two membranes होंगे, one outer and one inner. But what is different about plastids and mitochondria? The both membranes are going to be smooth. जैसे क्रिस्टे होते हैं, उनके पास कोई foldings नहीं होंगे. So again, this is also going to have the double-stranded DNA, circular DNA, and it will also have 70 as ribosomes inside it. So this is also a kind of similarity between the mitochondria and plastids. So plastids, as you know, these are also called as kitchen of the cell. So kitchen of the cell, you can specifically call a single type of plastid, that is the chloroplast. So but remember about the plastids में chloroplast होता है, that is kitchen of the cell. So this is present in plants and euglena only, बाकी cells में ये नहीं मिलेगा. The new plastids are formed by fission, यानी एक plastid break होगा to form two, and based on the pigment that is present in those plastids, there are three types. First is the leucoplast, second is chromoplast, and third is the chloroplast. Leucoplast, leuco means there is no color, that is it is colorless, that is no pigments. So no pigments, so these are present in non-green cells. So non-green है यानी कोई और color हो सकता है क्या उनमें? No, they don't have any sort of color. So generally जो white areas है या which are transparent, so these will have leucoplast. So these leucoplasts are majorly used for storage purposes. Right, leucoplasts are for what? These are for storage purpose. So storage, for example, if starch stored hai, in the case of potato or rice, then it is called as a myeloplast, because amylo is related towards starch. If it is fats or oils, as in case of oil cakes or oil seeds जो होते हैं like castor, then it is eleoplast. Eleoplast is going to be storing proteins, as in the case of maize. Then the second type of plastid is chromoplast. Chromo is the word that is related to color. So color है यानी instead of green, say colors like yellow, orange or reddish in shades. This is because of the carotenoid pigment. So chromoplasts have carotenoid pigments that can give different colors to the fruits or flowers or any other structures that have these kinds of tinge of colors. This is because of presence of chromoplast in them. The third plastid is the chloroplast, which is a greenish plastid, and it is going to have a photosynthetic pigment or a photosynthetic pigment that is chlorophyll. Because of this, the chloroplasts are called as kitchen of the cell. राइट जैसे माइटोकॉन्ड्रिया को पावर हाउस बोला है वैसे क्लोरोप्लास्ट को किचन बोला है बिकॉज यहाँ पे सिंथेसिस ऑफ फूड इज गोइंग टू ऑकर एंड मेजॉरिटी ऑफ द ग्रीन प्लांट्स आर गोइंग टू हैव दिस क्लोरोप्लास्ट इन देयर मेसोफिल region. So if you consider about a single chloroplast, since it is synthesizing food, it is of our more interest. So let us see in detail about the chloroplast. So chloroplast की thickness जो है it is about 2 to 4 micrometers. Length is 5 to 10 micrometers. Shape can be anyhow like a spherical, lens shape, oval, discoid, ribbon. It depends on the plant that you consider. Number it can be in Chlamydomonas एक ही होगा जबकि green algae में 20 to 40 हो सकते हैं. In plant cell they can even increase in their number. Then once you consider the structure of the chloroplast, so as I told you, chloroplasts are going to be double membrane. So one important thing that is you can say there are going to be two membranes, but these membranes, unlike the mitochondria, both inner and outer membrane are going to be smooth. So inner membrane is comparatively less permeable, जबकि outer membrane is going to be allowing everything to enter inside. So the space between the two membranes, the outer membrane or inner membrane के बीच की जो space है, so the in between space, what is the in between space called? The in between space that is going to be enclosed is the stroma. So there is going to be space which is enclosed. एंड द जैसे सेल के अंदर साइटोप्लाज्म होता है वैसे द क्लोरोप्लास्ट इज गोइंग टू हैव स्ट्रोमा इनसाइड इट and there are going to be thylakoids which is a stack of grana. So thylakoids क्या होते हैं? These are going to be membranous stacks and each stack का जो single entity है, the single entity is called as grana. So grana is going to be the plural form, whereas for the singular form you can refer it to granum. So what are these? These are kind of stacks of coins. So बहुत सारे डिस्क लाइक स्ट्रक्चर्स को एक के ऊपर एक स्टैक किया है और पूरे के पूरे स्टैक को क्या बोल दिया? Thylakoid. So that is the one thing to understand about chloroplasts. The second thing is there are many thylakoids. As you can see here, there are many stacks of grana. So हर स्टैक को that is हर thylakoid को कनेक्ट किया गया है by a membranous tubule which is called as stroma lamellae. For example, यहाँ पे एक stack है and there is another stack here. So two stacks are connected by a membranous thing that is called as stroma lamellae. So that is one another structure that you need to understand. And the membrane of thylakoids is going to enclose a space that is called as lumen. So again, जैसे पूरा का पूरा सेल कंसिडर किया तो सेल के अंदर के स्पेस में साइटोप्लाज्म होता है. If you take one organelle that is chloroplast, उसके अंदर के स्पेस को बोलते हैं stroma. But inside the thylakoid, if you see the space, that is called as the lumen. So that is again the terms that you need to understand. So now once we speak about the stroma, that is kind of similar to cytoplasm of the cell. So it is going to have many enzymes present in it, and these enzymes are going to function or for synthesis of carbohydrates and even the synthesis of proteins. So along with it, as I told, it has a double membrane like the mitochondria. It also has a double-stranded circular DNA. So this circular DNA is present, and it is also going to consist 70S ribosomes. So this is somewhat similarity between the mitochondria and the chloroplast. So that makes us to understand about the structure of the chloroplast. The next is about the ribosomes. So ribosomes, we have to understand about it कि इनको जो ढूंढा था, that was by George Palade, and since because of the name of the scientists, we also call them as Palade particles. Again, this is an important question, that what are Palade particles? These are nothing but the other name for ribosomes. These are non-membrane bound. Now this is a very important point. Which organelle is not having any sort of membrane? तो वो होगा ribosomes. जबकि single membrane हमने देखा है, then we have seen double membrane structures, and now that is no membrane structure. It is present in mitochondria and chloroplast as 70S ribosomes, and it is also present freely inside the cytoplasm as the 80S ribosomes. Now this is very important to understand, that is, it is a organelle inside a organelle. Now why is it called so? Because ये mitochondria के अंदर भी मिलते हैं. So mitochondria is an organelle and the ribosome is also an organelle. So organelle inside organelle, that is ribosomes. This is important. And what is cell inside a cell? Cell inside a cell is mitochondria. So that is the two things that you need to remember. Now another point here is about the polysome. Polysome yani poly is many. So many ribosomes which are connected onto a mRNA. mRNA is the messenger RNA. So messenger RNA के ऊपर जब बहुत सारे ribosomes attach होते हैं, then it is called as a polysome. So if you consider about the prokaryotic cell, that is the primitive cell, they are going to have 70S ribosomes. अभी 70S 80S बार बार नाम आ रहा है. What is it? So it is a kind of size denotion. So एक S जो लगाते हैं, that is the unit that is called as Swedberg's unit, which is a kind of measurement for the density and the size of ribosomes. So there are going to be two units for ribosome. Ribosome एक ही unit से नहीं बना है. It is made up of a larger unit and it is made up of a smaller unit. So this is going to be the units of ribosomes. So larger unit और smaller unit को जब साथ में combine करते हैं, you call it a single sized unit. So for example, once you are speaking for 70S ribosomes, it will have a larger subunit of size 70S and it will have a smaller subunit of 30S. अब दोनों को combine करोगे, so you might think that it should be 80S, but no, once they are combined, they form a 70S ribosome. Whereas if you want to make 80S ribosome, then you will combine a 60S larger subunit along with a 40S smaller subunit, forming 80S ribosome. So this calculation is not significant to understand maths, but yeah, that is the thing that you need to remember about ribosome subunits. अब ये subunits की बात की, that is one bigger unit along with a smaller subunit. अब इन दोनों को साथ में जोड़ के रखने के लिए there is a very important mineral that is going to be useful. That is nothing but magnesium. जब तक मैग्नीशियम आयन है तब तक ये राइबोसोमल सबयूनिट्स साथ में रहेंगे. They will be functional. The moment magnesium is deficient in the body, the subunits will depart from each other. वो एक साथ नहीं रहेंगे, and because of that the ribosomes will no longer be functional. So ribosomes का जो भी function है, that is to produce proteins, वो सब बंद हो जाएगा and the start of cell death will occur. तो जैसे बहुत सारे cells death होगी, even that is going to cause a disease. So magnesium is very important for bringing the ribosomal subunits together. The next is the nucleus, that is the big boss of the cell. It is all the control unit of the cell, which is going to have structures like nuclear membrane, nuclear pore, nucleoplasm, and nucleolus. Nuclear membrane, as you can see, जैसे सेल मेम्ब्रेन होता है, प्लाज्मा मेम्ब्रेन है, similarly nucleus is going to have its own membrane, that is nuclear membrane, which is going to have again two membranes, that is outer and the inner membrane. So जो outer membrane है, it is going to be continuous with the endoplasmic reticulum in the synthesis of proteins, that is the rough endoplasmic reticulum is going to be in continuation, whereas the inner membrane is going to enclose the nucleoplasm. Nucleoplasm क्या है? जैसे cytoplasm होता है, similarly nucleoplasm, which is a transparent semi-fluid colloidal solution, and the space between the two membranes, that is the outer and the inner membrane, there is a perinuclear space. So perinuclear space कहाँ होता है? It is not beyond the inner membrane but between the inner and the outer membrane. Then is the nuclear pore. जैसे pore है, it is kind of a open space. So this space is going to be the place wherein the outer and the inner membrane are going to fuse, and that particular fusion place is going to allow the proteins, the ribosomes, and the RNA to move in and out. So generally RNAs are to move outside, जैसे mRNA या rRNA होता है, वो न्यूक्लियस के बाहर जाते हैं. So these will move out from the nuclear pore. Then there is one big dense structure present inside the nucleus, that is nucleolus. So nucleolus, again, it is a non-membrane bound structure. So this is the second non-membrane bound structure that we have seen. The first one was ribosome. Second one is the nucleolus. So nucleolus also has the site for RNA synthesis, that is ribosomal RNA synthesis. So ribosomal RNA synthesis is going to be taking place not in the nucleus, but you can say specifically inside the nucleolus. The next is the structure inside the nucleus, chromatin. Chromatin जो होता है, it is not seen all the times, but it is majorly seen at the time of cell division. So cell division जब भी होगा, that is in the inner phase. Interphase में you can see the chromatins. Now what are the different phases of cell division and how does it occur? You can see in the other part of the video. So you can very well understand about the cell division. That videos are also available. Link will be provided down in the description. You can understand about the cell division as well. So these chromatins, how are they? These are loose extended network. So chromatin कैसे भी जाला बना हुआ होगा, and this जाला of the chromatin that has DNA, that is genetic material, histone, these are proteins, non-histones. So proteins that are histones and non-histones, these are for packaging, so that they can pack them very well and form the next structure, that is chromosomes. So these chromosomes, these are condensed structures. So when chromatin is getting condensed, so जब condensation होगा तब chromosomes बनेंगे. So if you consider the case of human cell, the human nucleus is going to form the chromosome, and these chromosomes is going to have about 6.6 into 10 raised to 9 base pairs long chromosome, that is about 2.2 meters of DNA. So you can imagine इतने छोटे से सेल जिसको आप माइक्रोस्कोप के अंदर देखते हो, in that 2.2 meters of DNA is going to get condensed and stay into the nucleus. So not even the entire cell is going to be available for this DNA, but just a small part of it, that is the nucleus. Now, another thing that we need to see is the structure of chromosomes. So, structure of chromosomes में the first thing that we see is it is going to be a kind of elongated structure. This elongated structure has two constriction sites जहाँ पे वो narrow down हो जाता है. So these constriction sites is one, the primary constriction site, and the second is the secondary constriction site. So these two जहाँ पे ये थोड़े narrow down हो जाते हैं, that is constriction sites. Now at the general center of the chromosome there is going to be a disc shape extra thing that is seen, that is the kinetochore. So this kinetochore is in the importance during the cell division जहाँ पे the spindle fibers are going to come and attach for the separation of the chromosomes. So at that time kinetochore is going to be an important structure of the chromosome. So there is going to be inside the chromosome the condensed chromatin is going to be present, और जो ending part होगा that is the satellite chromosome, and the end of the chromosome is called as the telomere. So that is the structure. The next part we see is the chromosomes. Once we have to understand kinetochore जो होता है, it is going to generally divide into arms. So kinetochore का जो location होगा, based on that you can say this is going to have a chromosome of a single chromatid. एक ही chromatid है, that is this structure. If it has two chromatids, that is the butterfly structure or the X-shaped structure. And one more term that we are majorly going to use in the cell division, that is homologous chromosomes. So homologous chromosomes, these are one coming from the maternal pair, the second coming from the paternal pair. So that is going to be understood in the meiosis, that is in the chapter of cell growth and cell division. So just to understand the diagrammatic form of it, that is single chromatid, two chromatids and homologous chromosomes, I have depicted here with a diagram. The next is going to be about the positioning of the kinetochore. So if the kinetochore is going to be present at the center, so center पे है यानी it is going to divide the chromosome in two equal halves. So that is equal arms, and because of it, once it gets pulled during the cell division, it will form a V shape. So as you can see here, it is forming a V shape when the kinetochore is at the center. If instead of being at center, it moves towards one side slightly and that becomes near to the center, तो वहां पे दो साइज के आर्म्स हो जाते हैं. एक जो बड़ा आर्म होगा that is the P arm, which is the longer arm, and the shorter arm will be the Q arm. The next is जब वो और थोड़ा अपनी सेंटर से भटक जाता है, near to the terminal when it comes, it will be acrocentric, forming a J-shaped structure जहाँ पे एक आर्म बहुत ज़्यादा लंबा हो जाएगा और एक आर्म छोटा रह जाएगा. That is the fourth type of shape of chromosome. And the last shape type is the telocentric, जहाँ पे telomere के पास, that is towards the exact terminal end, the kinetochore will be present, and because of that only a single arm is visible, that is forming a I shape during the cell division. So these shapes and the names, that is metacentric, subcentric, acrocentric and telocentric, is important to remember. अब ये chromosomes, how did the person look at it and how did he give the names and shapes to it? So generally staining is performed. So once you stain the chromatin or the chromosomes, there is going to be two regions that you can see. The first region is called as heterochromatin, second is called as euchromatin. So hetero is the word that means different, यानी ये जो region है वो बहुत अलग सा दिखता है because it is going to be darkly stained. वो जब आप स्टेनिंग करोगे, once you look into the microscope, this is very darkly stained, बहुत ज़्यादा डार्क होगा, जबकि जो other parts होगा वो बहुत lightly stained होगा, and you can see very light color, light stain pickup. So why did it stain dark? because वहाँ पे बहुत ज़्यादा density थी. It was very densely packed in that region, and once the chromatin is densely packed, वहाँ पे कोई activity नहीं हो रही है. Now what is the activity of the chromatin? That is to have replication. So replication नहीं हो रहा है, that means there is genetically inactive site. So that is heterochromatin, the darker area of the chromatin. And now euchromatin. Eu is the word that refers to true. Now true chromatin, because what is the function of chromatin? So that it is, replicating, or replication here, that is, it is genetically active. If it needs to be active, then it needs some space in between. So space अगर create होगी तो वहाँ पे stain accumulate नहीं हो पाएगा. So it is loosely packed, and because of loosely packed, it is lightly stained. That means it is genetically active for the replication process or for formation of proteins. It is actively functioning, that is euchromatin. So that was about the different structures present in the nucleus, and along with it we also complete the different organelles that are present in the cell. So I hope you understood what are cells, what are the different kinds of cells, and as well as what is present in detail about in the cells. So that was all about this chapter. Few more chapters are posted onto this channel. You can surely go and check them out as well. If you liked this particular video, then don't forget to share your love by like, share and subscribe. Till the next part, that is the next chapter, we'll come back soon. Till then, bye bye everyone. Thank you.